Aren't you curious how we got our parents' traits? Height, skin color, facial features? Well, that's because of genetics. But what is genetics? And who are the persons behind it? Together, let's find out.
Genetics is a branch of biology that focuses on studying genes, heredity, and variation in living organisms. It is a foundational discipline that explains how traits are passed from parents to offspring, and how these traits manifest in the physical and behavioral characteristics of organisms.
The foundational principles of genetics were first uncovered by Gregor Mendel in the 19th century. Gregor Mendel was known as the father of genetics, discovered the fundamental laws of inheritance through his experiments with pea plants. His work laid the foundation for understanding how traits are passed from one generation to another.
However, his work was largely ignored until it was rediscovered independently by Hugo de Vries, Carl Correns, and Erich von Chermach in 1900s. This marked the beginning of modern genetics. Hugo de Vries is often credited with rediscovering Mendel's work first. However, he initially did not give Mendel full credit, which led to some controversy. Next is Carl Correns. He explicitly recognized Mendel's priority in his own work, emphasizing the importance of Mendel's earlier discoveries. Then we have Erich von Chermach. He also acknowledged Mendel, though his work was more focused on plant breeding.
Next, we have William Bateson. He coined the term genetics and was a strong advocate for Mendel's work. Bateson also conducted important research on genetic linkage and inheritance.
We also have different scientists in the chromosomal theory of inheritance.
Walter Sutton and Theodore Boveri. Independently, they proposed that chromosomes are the carriers of genetic material and that they follow Mendelian laws of inheritance. This theory, known as the chromosome theory, was fundamental in understanding how traits are passed from one generation to another. Thomas Hunt Morgan, he demonstrated that genes are located on chromosomes through his work with fruit flies, or the Drosophila melanogaster. This discovery was crucial in linking Mendelian genetics with the chromosomal theory of inheritance.
Next, we have the discovery of DNA as the genetic material.
Frederick Griffith's experiments with Streptococcus pneumoniae showed that a substance from dead bacteria could genetically alter live bacteria, suggesting the presence of a genetic material responsible for inheritance. Oswald Avery, Colin MacLeod, and Maclean McCarthy identified DNA as the transforming principle in Griffith's experiments, indicating that DNA was the genetic material. Alfred Hershey and Martha Chase provided conclusive evidence that DNA, not protein, is the genetic material by demonstrating that DNA from bacteriophages carries the genetic information necessary for replication.
For the structure of DNA, we have the following scientists.
Erwin Chargaff discovered that in DNA, the amount of adenine is equals to the amount of thymine, and the amount of guanine is equals to the amount of cytosine, leading to the establishment of Chargaff's rules. These findings hinted at the base pairing mechanism in DNA. Rosalind Franklin and Maurice Wilkins analyzed X-ray diffraction images of DNA, providing crucial information about its structure and suggesting it as a helical structure. James Watson and Francis Crick, using Franklin's data and Chargaff's rules, proposed the double helix model of DNA. This model showed that DNA consists of two strands that form a helical structure, with a basis pairing specifically adenine with thymine and guanine with cytosine. This discovery laid a foundation for modern molecular biology.
Next, we have the genetic code and central dogma.
Francois Jacob, Jacques Monod, and Sidney Brenner discovered messenger RNA or mRNA, which transports genetic information from DNA to the ribosome for protein synthesis. Jacob and Monod also proposed the operon model to explain how genes are regulated in bacteria. This model is crucial for understanding gene expression in all organisms. Marshall Nirenberg, Hargobind Khorana, and other scientists decoded the genetic code, demonstrating that sequences of 3 nucleotides or codons in mRNA determine the amino acids in proteins. This discovery illustrated the central dogma of molecular biology, DNA to RNA to protein.
Then we have the advances in genetic engineering.
Kary Mullis invented PCR, a technique that allows for the amplification of specific DNA sequences. This revolutionized genetic research, enabling the study of minute amounts of DNA.
Next is modern genetic and CRISPR.
Jennifer Doudna and Emmanuel Charpentier developed the CRISPR or the Cas9, a revolutionary tool for genome editing. It allows precise modification of genetic material. Genetics in medicine has led to personalized treatments tailored to individual genetic profiles, including targeted therapies for cancer and genetic disorders. Scientists, among others, have collectively advanced our understanding of genetics, shaping the field into what it is today.